Good morning, good afternoon, good evening. Welcome everyone to our third webinar that we're running today on medical device packaging validation. Thanks everybody for dialing in and joining from wherever you're joining in from on this lovely afternoon. I'm going to be taking you through packaging validation. This is the third of a series of 4 which we've been on a journey for medical device validations. So today we are going to be specifically talking about medical device packaging validation. I will begin the presentation. So this is also being recorded. I'll give you a brief introduction about myself. My name is Adnan Ashbak. I'm your host today. I will be taking you through this presentation. I've been in the medical devices industry for 25 years. And from those 25 years, the first, I would like to say 12 years was predominantly working through validation, equipment validation, largely packaging for medical devices of various different types of natures, different classifications, which hopefully I'll be able to apply some of that knowledge and share that with you today. So without further ado, I will now start. What we have is, as I said, this is the third of a series of webinars that I'm running, and the first 4 specifically are on validation. So on the 24th of September, we discussed process validation as a basic overview, and then on the 16th of October last month, we discussed IQ, OQ, and PQ in a bit more detail. Today we'll be running through packaging validations, which will also be looking at sampling and statistical validation and sterilization validation, but very briefly. There's a lot to cover in these areas. It definitely doesn't do it justice to cover these 3 big areas in 1 hour. So we will be predominantly talking about packaging validation, and then next month, the 12th of December, because we're getting close to Christmas, I'm sure people will be thinking about other things as we get closer to Christmas. We'll be looking at equipment software validation. And then going on to 2025, we will be exploring further subjects in our medtech journey in the medical device development. We will be looking at ISO 13485, the EU MDR, IVDR regulations, software as a medical device, manufacturing requirements, design control, risk management, FDA regulations, sorry, that's a typo, clinical evaluation, validation and verification testing, and startup essentials. So with that said, let's move on to our program. So we've got just under the hour. I will try to allow for 10 minutes for questions. So it's a bit of a challenge ahead for me. So I will begin without further delay. So let's start off with what packaging validation is. So what packaging validation can be defined in many different ways, but essentially what we're looking to do in packaging validation is ensure that the packaging of the medical device is effectively protecting the product, is preserving the product throughout its shelf life, is maintaining its sterility, that's of course if it's sterile, and of course complies with appropriate regulatory requirements. So the importance of packaging validation can be to reduce the risk of damage to the device during transportation and storage. It's also to prevent packaging failures that could compromise patient safety, reduces the risk of loss of sterility and preventing contamination. Of course, we have to ensure compliance with regulatory standards. These regulatory standards, sometimes they will give us enough guidance to be able to achieve an acceptance criteria or ballpark that we're looking to try and achieve. And of course, defective packaging can result in significant costs on medical device manufacturers, which can affect reputation. So we don't want to get into that position. Some of the regulations and the standards that we have to look at, so these are regulations. So 21 CFR Part 820, of course, for the US FDA, Subpart G, talks about packaging materials to be compatible with the device to meet safety, sterility, performance requirements, and packaging and related process must be validated. And then section 250 then requires manufacturers to validate any changes to packaging materials or processes that could affect the safety or performance of the device. ISO 13485 then in various clauses talks about similar things, looking at safety and the functionality of the device and making sure that any changes are traceable and they're managed. And then ISO 11607, which is the standard for terminal sterilization for barriers, is looking at material properties and looking at the sterile barrier systems. And then the amendment in 2023 is also looking at expanding the application to risk management. So there's been a few revisions in that, and then also looks at details the validation of packaging systems, including sealing equipment and processes, and then also outlining the requirements for storage and transportation. Then the EU equivalent of the FDA, the European Medical Device Regulations, also looks at and defines in Article 10 that the manufacturer's responsibility is to validate packaging processes and provide technical documentation demonstrating compliance with applicable standards. And then, of course, it's similar again, where its definition or its aims and objectives is to ensure that the packaging is protecting the device throughout these different phases of transport, storage, and handling. So ASTM F1980 looks at guidelines for testing and validating the shelf life and sterile barrier systems, including how aging should be accelerated under control systems. And that then helps manufacturers predict the durability over time and ensures that the sterile barrier systems remain intact throughout the product's life cycle. And then ASTM F209723 provides recommendations on design, development, and evaluation of primary packaging systems for medical devices. And it also helps ensure that the packaging doesn't affect the device's functionality or safety. There's lots of other substandards. So there's 868-5. There's also a -6 as well. So we have to have a look at these in conjunction with ISO 11607, which then allows us to look at different types of testing, but seal width testing, seal integrity testing, and so forth. So these are guidelines specifically for heat sealable flexible packaging systems. And then, of course, there's different types of sterilization which the device would undergo, which could be ETO, ethylene oxide, or gamma radiation. These are the two popular ones. And that's also got to be considered because there's also an effect that the radiation can actually have on the seal. In some cases, the over radiating the seal can have an effect on over sealing or strengthening the barrier, which causes further problems because we want to make sure that the sterile barrier is intact, but the seal strength doesn't go beyond a certain limit and it doesn't go below a certain limit. So these standards are here to define some of those parameters and acceptance criteria. And then ASTM D4169 and the ISTA standards, the international shipping transportation, they are there to ensure that the packaging is intact for shipping in the various conditions, for vibration testing, for impact testing, for compression testing, for negative altitude if you're in the air, if you're transporting by shipping, by road, rail, etc. We have to consider the entire journey of the device, and these different levels within ISTA would then dictate what is required. Now, I've seen some medical devices undergo ISTA 1, which I would definitely challenge because it's basic test for prototypes, and it's so basic that it wouldn't even meet the requirements for basic medical device testing. So therefore, ISTA 3 is usually the recommended starting point for validation of packaging, which then leverages ASTM D4169 and the subsets, the different dash subsets of D4169, which then go into the different levels of testing which is required. So moving on to packaging design. So before we even get anywhere near testing, of course, the design of the package has to be compliant with the type of device, which of course is going to be risk averse, which we'll come on to in a few slides. So looking at the three different levels of packaging, primary packaging, secondary packaging, and then tertiary packaging. Well, primary packaging could be defined as the immediate packaging that comes into direct contact with the device. That would then serve as a sterile barrier for the sterile devices. So examples are blister package, pouches, syringes, which are containing the medical device directly. So it's in contact directly with the device. And then you've got secondary packaging, which could be the outer packaging containing the primary packaging, which has extra protection. Sometimes that can also be sterile, so there could be a double sterile barrier that's required. In this case, these examples are corrugated boxes, shrink wraps, etc. But often that might not be the case. It might be the case that you've got something similar to a primary package, which is the secondary barrier, and then the tertiary packaging is in fact the corrugated boxes. But in this case, for this example, if we follow this through, then tertiary would be your packaging for bulk or palletized shipments for transport and handling. So typically large scale distribution. So here we've got things like pallets, wooden crates, stretch wraps, those type of things. So again, the design of the packaging should be based on the nature of the device itself. So we're going to look at this risk-based. That's what it really boils down to. You're going to look at the risk of the device. So the type of packaging that's been chosen is really got to think about the risk of the device that's posed if you leave the device exposed or it's less protected, etc. You know, it's really, well, of course, dependent on the actual device itself. And we will look at some examples. So the packaging must be tailored to the shape and the size of the device. Some devices could be sterile barrier, must be compatible with the sterilization method used, of course. So again, if you're using gamma radiation or ETO sterilization, you're looking at the permeability of the gases through the packaging, that it can actually kill the bacteria that it's aimed to do. So the microbial count that it's looking to reduce to ensure that the sterilization method is successful. So when we're looking at sterilization validation slides, we'll come on to that. So some devices may be sensitive to factors such as moisture, light, temperature. So again, the protection has to be appropriate. Sterility, integrity, and functionality must be maintained for the entire shelf life. So again, that needs to be tested. So there's no use in choosing packaging materials which will not withstand the entire shelf life. So whether that's 2 years, 5 years, or whatever the device requirements are. So for example, if you've got implantable devices, such as a pacemaker or a stent, typically packaged in rigid containers, you'd have a sterile barrier. In some cases, these are vacuum packaged blister packs, a smaller device. Because devices are sensitive to physical impacts, the packaging must resist compression and remain sterile until its use. And then when you've got things like diagnostic kits, pregnancy tests, and blood glucose meters or monitors, typically, again, they could be in blister packs, folded cartons with plastic boxes, and then these could also be in different compartments. And then also, again, because you've got moisture and contamination, which can reach the device, that needs to be protected. So the purpose of the blister packs has to be to protect also from moisture as well as contamination. One of the other things to consider also is when you're looking at the packaging design is that you may also have the instructions for use or labels or any additional materials that will be packaged alongside the device itself. Then that also needs to be considered as well as part of the packaging. And when we come on to the actual packaging design for the shipping testing, you also need to include that as part of the tests. So when you're designing it, ensure that it's going to be compliant for the testing requirements for the ASTM standards. For sterile devices, the design must include an effective sterile barrier to maintain the sterility. And then labels should also be very clear. They should be readable or legible, accurate, and they have to comply with regulatory requirements. So there are ISO standards for the requirements stipulated for the symbols that are required on labels and the instructions for use in the packaging. So when you develop the initial packaging prototypes, they should reflect the final design as much as possible. So when you're doing the validation and testing, it should be as close as the final testing as possible, or at best, it should be on the final design. And that includes the materials, the seals, and the labeling. So of course, if that fails and you change, again, it could be because you're still under design and development, you've not got to design freeze, you're still trying to identify whether this materials is appropriate. If you've got a situation where you're retrospectively validating, which is very often the case, and you're not basic design and developing, something may have been picked up during an audit that you haven't had adequate packaging validation done. You can also look at historical information and the legacy of the device. If you've never had any complaints to do with packaging, that will probably give you a good starting point that the devices materials that have been selected have probably been appropriately selected, but you need to then take it through the rigor of testing. However, if it fails the testing, that's when you need to think whether the design is accurate and robust enough, because the proof really will be in those tests. You can try to use and leverage some historical information, but in front of an auditor, if you've got failed data, which is coming from the standards not being met, that's going to be a hard argument to try and hold. So other things to think about when you're selecting your packaging material, it to be compatible with the device and the sterilization method, if applicable. Now, obviously, again, that's assuming it's a sterile device. If it's non-sterile, then you should look at the appropriate compatibility, but not with the sterilization. Making sure that you're providing adequate protection against damage, contamination, and other environmental factors. Some of the things that you should think about is where the device is to be sold. Now, if the device is to be shipped in a subtropical country or climate, then you should really be thinking about those temperatures and the humidity that's being reached in those areas and regions. And the same thing if it's going into a sub-zero temperature climate, you should also think about the factors of the environmental factors that the device will be exposed to under those conditions. And then also looking at levels of extractables and leachables. There's also the possibility that materials could extract into the actual medical device and damage the medical device and increase the risk. So you have to make sure that the materials that you're choosing don't leach into the device and they're safe to use for that purpose. Also be for biocompatibility testing, which come into direct contact with patient if necessary, and that's one of the important reasons for looking at extractables and leachables. So some of the things that we, as I mentioned earlier on about labeling, these are just some examples. That's not an exhaustive list of labels symbols that you should be thinking about on the label. So if you're selling in the European Union, then you need to think about the CE mark. If it's a class 2A and above, then you need to think, or even if it's a class 1 sterile or measurable or reprocessing, then you need to think about the notified body number, the NBOG number with the CE mark. Expiry dates, lot numbers, if there's a biological risk, if it's non-sterile, if it's sterile, the serial number, caution, single use, keeping dry. These are just some typical symbols that would be used, and you should refer to ISO 15223. That's 223 or 23? Yeah, it should be. That's the typo. ISO 15223-1, not 15233. That should be 223. And also the medical device symbol, the MD symbol. There are also some other introduced because of the MDR. There were some other symbols that were introduced. Specifically, if you've got an implantable, there's an implantable card that you have to introduce, and then also there is another ETO or sterilization symbol that's also been introduced as a result of the MDR. Again, if you look at MedTech Europe, you should be able to find some guidance on there as well. And I think it's been introduced now into 15223 and other ISO standards as well. But this is something that came out during the MDR. So think about the symbols that are also required for the type of device that you're applying this on. So now moving into sterilization methods. I talked about ETO, ethylene oxide, and gamma radiation, which are very popular. But then also you've got the possibility of electron beam, E-beam, ozone, ultraviolet possibly, autoclaving, dry heat, different methods that will be used depending on what the level of bioburden is that's being reduced within the device's packaging, and it's to maintain its sterility and safety. So a number of things that we need to think about there. And then specifically, if you're looking at gamma radiation, look at ISO 11137, and that's looking at where you've got high energy gamma rays, which is used from a cobalt source, and it's used to kill the microorganisms by damaging the cellular structure. And it's widely used in examples such as surgical instruments and catheters. Advantages are that it's highly effective against a range of microorganisms. It's fast process. It can be suitable for heat-sensitive packaging, deep penetration for thicker packaging. Limitations are that it can cause material degradation, may affect the packaging's integrity over time. And also, if you've got a cobalt facility, there's also risks to humans because of gamma radiation. So there's lots of things to consider about in packaging, whether it's something that you should really go down the path of. So there's different considerations, advantages and limitations to think about. Now, ETO, which is commonly used in other devices, such as for plastic components or electronic devices, ISO 11135 is the standard that talks more about the requirements for ETO sterilization. So that's a gas which is used to sterilize by disrupting the DNA of the microorganisms, preventing the replication. It's used for heat and moisture sensitive packaging. So that's where you've got things like Tyvek packaging and permeable, gas permeable packaging materials, those type of materials that are used. So advantages are that there's a wide range of sensitive materials, including those with complex geometries that don't require higher temperatures, such as the gamma radiation is needed. It's also deep penetration. Limitations are that there's longer cycle times, there's potential toxicity, and there's need for aeration to remove residual gas, and there's also material degradation to think about as well. However, depending on, again, the construction of the device and the requirements for the device, it may be the suitable go-to packaging that you choose. So when you start thinking about writing up documentation for validation, you should always begin with a validation plan. Now, in the previous session, I discussed IQ, OQ, PQ, and the requirements of these different documents. I also touched upon the validation plan. But for the benefit of the audience here, when you're writing a validation plan, in the same way for packaging validations, you should begin with a typical structure that looks something like this, where you have the purpose and the scope, which defines the goal of the validation, what you're trying to, what's the device that you're trying to protect, describe what areas of the packaging are being validated, there's different materials, whether steel integrity is a thing, the different regulatory requirements, look at the standards, the design components, specify the materials used, example, the films, the trays, etc., and then look at the packaging attributes, barrier properties, durability, and then also looking at risk assessment, outline the potential risks associated with the packaging failure, what sort of things can go wrong. So you may have already done a risk management exercise at this point, or you may not have, but then at this point you should then think about looking at the risk scenarios related to the device and its exposure and things that could go wrong with potential failures, and then identify how you would be mitigating that risk by the design and the improvements of the testing that you're going to perform. The testing that will be done for the packaging validation specifically is there to mitigate risks. which could be to do with seal integrity. That seal could be compromised. It could then cause bacteria to get into the device, which is otherwise sterile, and it could then cause the device to become non-sterile. That could then lead to bacteria in infection in a patient, which could ultimately lead to death in some cases if the infection could be so serious, especially with things like implants, where you've got biological materials which are being protected in packaging, which the sterility is such a high factor and the integrity is so critical. Any breach of that barrier could then lead to bacteria being spread within the implant, which could then be transferred to the patient and then could cause lots of severe catastrophic issues within the patient, and that could lead to death. And then looking at pass and fail criteria, what for each testing, sample size and selection, specifying the number of packaging units, how many, why you select that, what's your rationale, defining the testing conditions, where will you be doing this testing. So that could be in a clean room, and we will talk about clean room in a few slides, because that's also a critical part of packaging validation. So other things to think about also in your validation plan could be equipment, materials, what materials you're using, what equipment you're using, equipment for testing, define the specification of the materials. When it comes to equipment, you could be using compression testers of environmental chambers. Are you doing it in-house? Are you doing it outsourced, as in an external laboratory? Or are you doing some of these tests in-house? Now, if you're doing the tests in-house, there's also a caveat. There's an assumption that the equipment that you're using has been calibrated, is gone through some level of maintenance, and maybe even the equipment itself needed to be validated. You then have to list out, of course, roles and responsibilities, who's responsible for which task, and any training that's also needed to be executed before doing the packaging validation. So writing up the plan, and then before doing the testing itself, after the plan's written, is there any training that needs to be executed before actually performing the tests? And then also, what's your strategy for change control? If you need to change the packaging materials in the midst of the packaging validation itself, maybe because you've identified that the materials are not right, you need to go back and make some tweaks. This is very much in a R&D situation where you're prior to design freeze, right? So you're not in the design freeze stage yet, so you're needing to think about these type of things. And then also acceptance criteria and conclusions. So what sort of things that you need to abide by, and what the conclusions are of the overall testing. Oh, I think that's skipped. Yeah, right. So now moving on to risk management. Right, so risk management, as I said, I've mentioned risk a number of times. Why do we perform any validation, whether it be design validation, packaging validation, whether it's clinical trials, whether it's usability, any testing that we do is always coming back to risk, to ensure that the device is safety for the patient is paramount. So packaging validation, of course, isn't any different. I've also already outlined some examples of severities which could lead to death because of poor packaging or packaging breach or inadequate testing that's not been documented, not been performed, which could lead to death. So it's really, really serious. So I really like to make sure that the audience here understand the utmost importance of carrying out packaging validation, especially when you've got a sterile device, and even more so when you've got a class 3 device, which could be an implant, like I've explained, those severities and the breach of the packaging and what that can lead to. So when you're looking at your risk management, it's identifying the hazards, consider all the potential risks of the life cycle, such as contamination, material failure, degradation of packaging over time. Right, so it's great having a device which is it's integral and it's maintained its integrity at this moment in time. But what happens, say, 15 months from now, if the label says that it's got 2 years of shelf life and that's 24 months, but in between that time, if there's a breach that happens and it fails because of that, it means that we didn't do the testing adequately over a period of time. So it was done at a moment in time, but not proving over time. And that's why aging and shelf life trials is so important as well, and to make sure that you have a stability chamber which is calibrated and integral, that you can put the device with the packaging in and test it over a period of time. So there's a few slides also coming up just about stability as well. So as I said, risk management, identify, do your risk analysis. Some of the things that potential risks: chemical migration for packaging materials through the device, contamination due to improper sterilization, risk of toxic exposure to patients from residual sterilizing agents, loss of function of the device, physical deformation of the device, degradation of the device, punctures or tears breaching the sterile barrier, crushing of the device over compression. These things are real. You know, I've had these experiences. This is just an example. Some people do things differently. Unacceptable, acceptable. What is your criteria for acceptability? Some people say no yellows, only greens. I would probably these days favour having a green and a red, just having a very clear distinction between what's acceptable, what's unacceptable. But if you have a risk portfolio for severities of 5, the probability of 5, then ultimately you don't want to be in any of these reds. You want to be in the yellows or the greens, and then that would determine your risk acceptability. So when you've identified the risks, make sure that the measures can be implemented, and the whole point of implementing these mitigation measures is that they are there to reduce the risk. So the risk that's been identified, that it reduces the risk. So if you score something like a 25 or a 20 or any of these reds, as most people know, that the objective of risk management is to get it into the greens, into an acceptable level, to reduce the level. So you want to see if you can reduce the probability. The severity could be difficult to move, but the probability, likelihood of that risk happening, because you've introduced the mitigation measure, would then bring this back into a control which would be acceptable. So looking at design controls, looking at process controls, and looking at sterilization compatibility. So looking at the design, choosing the right material. Implementing quality control systems using the manufacturing process, such as automated inspection systems for seal integrity, ensuring that the packaging material is compatible with the sterilization method, which is, I've said that over a few times. So statistical sampling, just moving into statistical sampling briefly. Now, as I said to you before, there's a whole masterclass that you can do on statistical sampling, and there's lots of the other courses like green belt and black belt courses that you can do on this. We're certainly not doing that in the next few minutes. So on a high level, when you're looking at statistical sampling, some good standards to use are ISO 2859 and 3951. So 2859 would be looking at more your attribute tests, and 3951 would be looking at your variable tests. There are also the equivalent ANSI Z1.4, I think it is, which is the American equivalent. And also there's a BS standard as well, which also looks at similar sampling plans, but I prefer to use the ISO standards, and that's what I've used in the past. Typically what you do is you would have a look at, these came from the military standard originally, and there's tables within the standard which on a sliding scale allow you to look at the lot size of the batch. And then based on the batch, you would then be able to, using that sliding scale, define a number of units that you would take as samples during your manufacture and test it for its packaging integrity. So this is where you would apply something like AQL, so the acceptance quality level. Sometimes there's, if anyone knows of that, I don't have the chart in front of me, but the table itself will have different levels of acceptance quality level. So 0.65, for example, would be a norm that would be used. So if you move towards the right, which is a higher acceptance quality level, that's assuming that you've got a higher level of confidence. And if you move to the left, it's a lower level of confidence. So 0.65 would probably be something that you would have a fairly stable process and you have some confidence in the process itself. If you have a brand new process, it might be better to tighten that up and look at the acceptance quality level to be a bit more tighter, so that the number of samples that you take may be more stringent. So looking at taking more samples from the lot. The whole point of using statistical sampling is that you're not destroying an entire batch for testing. So you take a representative sample size from the batch, which if it passes the acceptance criteria, then you can assume that it's passed the entire batch. If it fails, then you assume that it fails the entire batch. And that's the whole premise of using statistical sampling. So, so defect would be a single non-conformance or deviation from the specification. A defective unit could be a unit containing one or more defects. And then you also have to apply the inspection levels, defining the severity and thoroughness of sampling, which is also based on device criticality. So when you're looking at defining statistical sampling, you define the population size, the lot size to be taken, the number of samples. Larger batches require larger samples. Choose the AQL, so based on the severity, critical, major or minor, and the impact of the defects on the patient safety and the device functionality. And then a sampling plan is selected based on the AQL and the lot size. So the plan will then give us the sample size, it will give us the acceptance number, so how many you have to achieve to be acceptable, and then what's the rejection number. So what it will do is typically say that you might have a sample size, or let's just say, for example, argument's sake, of 50. Within the 50, there will be a table within the standard, and the table will then tell us, based on the AQL, if we use 0.65, for example, level 2, let's just take, for example, so we talked about the levels, the inspection levels, level 1, 2, or 3, and if we use level 2, which again is a sort of nominal value that's used, we would then accept on, say, it may be 49, and then if it's failed on 1, then the entire batch has failed. So even one failure out of the 50 would represent a failure for the entire batch. So this is very, very important when you're looking at the rejection numbers. I think I'm going to speed up a little bit here. So looking at the inspection methods, so looking at attribute and variable sampling, so what type of testing that you have to do. And then looking at, now moving on to clean rooms, just quite swiftly looking at this. So ISO 14644-1, -2, and -3, these are the standards that you need to have a look at. So what type of clean room or control room are you packaging in? Or maybe at this point you have not even decided, but you need to decide based on the cleanliness that's required for the device that you might be developing at this stage. And there's often questions, how clean is clean? And you need to then look at the risks of what cleanliness you need to achieve. So based on that, you will then be able to determine how clean the environment needs to be. Does it need to be on an ISO class 1, which is very high, class 2, class 3, the different classifications, which represents bioburden levels in the environment, which is acceptable levels, the number of particle counts per cubic meter that's acceptable. So for example, an ISO class 100,000 in the old terms represents 100,000 particles per cubic meter that you're allowed to achieve, or 10,000 or 1,000. So these are the different classifications. So the lower the number goes, less particles are acceptable, the more controlled and clean the environment. So this is very important, looking at classification of clean rooms. The air quality has to be controlled. You have to then look at environmental monitoring, temperature and humidity, and then looking at PPE, what sort of, do you need to wear gowns, masks, what rigor needs to take place within the clean room. When you're designing the clean room, look at the classification, the cleanliness level, choosing the appropriate location, installing air filtration systems, setting up controlled areas, having protocols for personnel equipment when you're coming into the clean room, when you're exiting the clean room, what sort of control measures need to be in place for that. So the clean room itself needs to be validated. So have you reached the required levels of testing? So this is where in the clean room testing, you would then do air particle counts, measuring the concentration of the particles in the air flow. So there can be different times of the day. Also, the airflow and the particle count can be affected by body movement coming into the clean room and traffic coming into the clean room. So you've got to look at airflow and air changes, looking at the air velocity, looking at direction, the volume of the correct flow, pressure differentials that can also be affected, and temperature and humidity can be affected by the pressure in the external environment. So even in the atmosphere, if it's a wet, damp day, it can affect the pressure differential in the clean room. And I've had situations like that, having also worked in the pharmaceutical industry, where we've had to put placebo powder into a bottle as part of packaging validation, where it's been a damp day. It's been absolutely impossible to try and control the volumes to make it placebo. It can look as if it's expanded, the powder, but the actual weight of the powder is exactly the same as of a bottle that looks more and one that looks less. And the reason for that, again, when we did some troubleshooting on this, was because of pressure differentials. So I'm not going to talk too much about design qualification, because we looked at qualification during the last webinar. We looked at the DQ, we looked at IQ, OQ, and PQ. So I am going to skip and fast forward some of this. The requirements are the same. I mean, I do recommend you to have a look at that video, which is on the Simply Medica YouTube channel, and you can see some of the requirements that are generally required for DQ, IQ, OQ, and PQ. Making sure that the protocols are there, making sure that you've tested the extremities, and making sure that when you get to PQ that you are documenting everything in a close to the real environment as possible, and documenting everything as such. Process validation also in sealing packaging. This is just some examples to make it more meaningful for you. What sort of things that you might look at in packaging validations for IQ, OQ, and PQ. So you might be looking at, so the machine set up correctly, is it the machine connections correct? Are they fitting horizontally? Is the correct machine supply, the electricity, power, OQ? You're looking at functional testing, sealing temperatures, sealing time. So you're looking at the high temperature, you look at the low temperature, you look at the high time, you look at the low time, and then when you get to PQ, you look at real world conditions, sealing temperature, which would be nominal. So in this example, you've got 160 and 180 at the low, and then 170 could be the nominal temperature. And then also looking at the dwell time, which is the amount of time that the pressure plates that are coming in contact with the seal when it's sealing the packaging is 4 seconds in this case, which is in between the high and the low. So then when you're getting to the testing, you do seal strength, look for adhesive lines, foreign substances, you do dye migration, and you compare that against acceptance criteria. So in the test protocols, you want to be looking at things of visual inspection. You want to be looking at things such as packaging, such as cracks, defects such as punctures, contamination. You also want to be looking at the seal integrity, which is really important, and you have to look at things like the tensile strength test, applying tensile force, and then measuring the amount of force that's required to break the seal, which is usually measured in newtons force. You then look at the peel strength, to measure the force that's needed to peel apart the sealed edges, and then a dye penetration test. You would use a dye to check for leaks in the seals, and then a bubble leak test when you submerge the sealed package in water, and then you check for bubbles. The standards that I mentioned earlier on will give you guidance of some of these tests and the acceptance criteria of what the benchmark testing is for these barrier tests. And then you've also got barrier properties testing, which is water vapor transmission testing. You've got oxygen transmission testing, particulate contamination testing. This is when you're looking at possibly class 3 medical devices, which really need to have a very high benchmark, and the testing that's going to be done, the specification or the acceptance criteria of the micron level of an acceptable particulate would be really low, and you would then need to look at some of these much more critical tests of the packaging. When you're looking at the transportation testing, we talked about the ISTA and the ASTM D4169. You're looking at drop testing, compression testing, and vibration testing. That tests for physical performance testing. Then you're looking at also the aging testing for the shelf life. You would do accelerated aging testing and then real-time aging testing. So accelerated, as you may imagine, is exposing the temperature over a faster period of time. So it's simulating that time, but much faster. And then real-time, as the term suggests, it is in real time. So if it's 24 months, 2 years, then you've got to then store the samples in normal conditions over 24 months and test it periodically for degradation. And then other tests that you might want to look at also for sterilization testing are pre and post sterilization testing, thermal stability testing, microbial barrier testing, and bioburden testing. So also looking at the bioburden levels. And then also reviewing the content. So if you've got labeling, making sure that it's legible, that you can read it under different conditions, that the information is also accurate and appropriate for what is required. So ISO 11607 also in section 7 talks about usability as well. So this is really important to look at the usability of the device, because it's all very well designing a device which is very strong in its protecting its contents, but it's so difficult to open the device that you can't even get into. So this symbol, for example, here is a universally accepted symbol to identify where to open from. So open from this side, peel open, and this symbol here, sterile unless package is damaged or open. These are just some examples that are used. And then looking at 7.2, when you do your usability evaluation, you should look at the ability to identify where to begin opening, what the technique is to open, the ability to subsequently present the context aseptically if it's required. So section 7 looks at the usability. Here's some test equipment that you would be using or your external laboratory might be using. So tensile strength tester, environmental testing would be for aging testing, compression testing for shipping testing, bubble leak testing, drop testing, water vapor testing. This is some examples of equipment that would be used in the whole process of packaging validation. And then also vibration testing, altitude testing, and label abrasion testing. So this covers everything that we talked about. This is the type of equipment that would be used in the process of ensuring that your package has been validated. So some of the things that we're looking at for acceptance criteria is that making sure that the seals pass a predefined peel strength or seal integrity test, that there's no breaches in the sterile barrier, no adverse effects to the device, no deterioration or degradation after the environmental testing, labels are legible and intact, include critical information, no deformation, leakage, or damage to the packaging, no detectable microbial growth or contamination inside the packaging, no deterioration, degradation, or damage after accelerated aging testing, and no chemical or physical reactions between packaging materials and the device. So when you've done all of that, of course, you've got to document everything, have a validation summary report, which looks at risk assessment. You've documented test protocols, you've met the acceptance criteria, you have the results, and then you've also looked at corrective actions where required, and then making sure that the documents are met through regulatory compliance. So somebody who's in your QARA department would be ensuring that the correct standards have been followed. So just sort of final example on foil pouch validation, you would outline packaging details, outline the test methods, outline the acceptance criteria, and then perform, test, and write up the report. Final design review. So when you look at the design review after design freeze, you would be looking at the design inputs and the requirements, review the packaging production process, testing and validation overview, review the outputs, review the risk management to make sure everything has been documented from a risk perspective, and all of the necessary documents are then in place. If you need to revalidate, there may be a requirement to revalidate on a periodic basis, and then also looking at complaint handling and corrective actions. So I'm going to summarize. It's been a very quick hour. So looking at, always think about the user requirements. So if the device is sterile or non-sterile, you know, your packaging outputs are going to be different. Think about the risk that the device is exposed to by poor packaging or a breached seal. Design the packaging to minimize risk, but thinking about the user. Ensure you're using the correct symbols on the packaging. Ensure that the equipment used for testing is calibrated, maintained, and validated. Ensure the environment in which the packaging takes place is appropriate also to risk, such as if it needs to be in a clean room or a controlled room, or is it okay to be in a dirty room, assuming it's non-sterile at that point. Ensure controls are reflective accordingly. So do you need to have PPE? What sort of equipment, gown, what procedures do you need to have? Face masks, all the rest of it. And ensure that you've got the correct resource to complete these activities. So it's ensuring, you know, looking at all of this in as a whole. So I'm just going to wrap up. We're near the hour. I'm just going to open up for any questions. Got the last few minutes for questions. I have one question in the chat box. So I forgot to mention you could put it in there's a Q&A chat box. I have a few questions below. Number one, is it mandatory to do packaging and transport validation for non-sterile device class 1 per FDA, EU, SFDA? Right, that's a good question. So the thing is, it's all very good practice to do this. Now, if it's a non-sterile class 1, according to these regulations, you may not be reviewed by a notified body to do to whether you're performing these activities or not, but it's definitely good practice to do that. Now, assuming that the regulatory authorities may look at the technical documentation, then it's still a requirement. I mean, the safe answer is that you should be performing transport validation regardless of classification. But what you need to look at most importantly is the risk. Assuming it's a class 1 and a non-sterile, that the risk would be minimal. That would then dictate what level of packaging testing you would need to do. So at this point, I mean, it could be quite minimal testing. So you might still do some transport validation, but think about the regions where we would be transported to. Now, you could also look at a justification, but you have to then look at this risk-based. So you could write up a justification, that's possible as well. You've also asked a second question here. Can transport validation report per ISTA 2A utilized for FDA registration for devices? Yes, that's also possible based on the rationale for you selecting the different levels of ISTA. So whether you even chosen ISTA 1, but I've spoken to test houses before, and they don't like ISTA 1 being tested for medical devices. It's just not common practice. ISTA 2A might give us more testing, but ISTA level 3 will definitely give us the requirements for medical device testing. Any other questions? I think if not, then we can wrap up. I don't see any other questions in the chat box, but I'd just like to thank everyone for attending once again. And if you haven't already subscribed, please do so. And you have a look at our website, simplymedica.com. If there's any assistance that you need, please reach out.